Hi, everyone. I think we're going to get started. It looks like some of the requests to join have died down a little bit, so we'll get going. My name is Corey Nelson. I'm the Director of Development in the College of Sciences at UNLV. I hope you all can see my screen. If not, I'm just going to go over some of these rules for you really quickly before we get started. Also, if you are having any difficulty hearing, please let me know in the chat, and I will try to see if I can figure something out. But first of all, thank you for joining us again. It's such a pleasure to have you here at Virtual Lab Notes. This is the second webinar that we've done featuring some of our wonderful faculty at the College of Sciences at UNLV. Just as a reminder, make sure that you mute your microphone and keep it muted for the entire presentation. And also turn off your camera or video and make sure that that is also not going as well. It can slow down the presentation if those are on. Make sure they stay off for the entire presentation. Second, utilize the chat function on the right-hand side of the screen to ask questions. We'll save about 10 to 15 minutes at the end of the presentation for Q&A. If a question pops up while Dr. Strong is presenting, feel free to pop it in the window and I will write it down and make sure that it gets asked at the end of the session. If during the presentation you encounter any issues, you can access the Google Meet Help Center at support.google.com/meet. That should be able to answer any questions you have while the presentation is taking place. And lastly, we will have today's presentation. As you can tell, it's being recorded. We will have it available for viewing on the website at unlv.edu/sciences/virtual-lab-notes. And you can see last month's presentation from Dr. Brian Hedlund at that address as well. That page is up and running. I'll put the link in the chat box just so you have that available also. Before we get going today, though, I just want to let you know that we will be doing another one of these sessions in July. So Wednesday, July 29th, please join us as we hear from Dr. Ernesto Abel Santos, who is a professor in the Department of Chemistry and Biochemistry in the College of Sciences at UNLV. He will be presenting on prevention and treatment of infectious diseases in the COVID-19 area and beyond. So mark that on your calendar. You'll get email reminders about it as well. And before we get going today, I do want to take a moment to introduce our presenter, Dr. Christy Strong. Dr. Strong earned a BS in biology from Montana State University in 2001, an MS in biology from East Tennessee State University in 2005, and a PhD in integrative microbiology and chemistry from the University of Montana in 2011. After completing a postdoctoral fellowship at UNLV under the mentorship of Dr. Martin Schiller in 2014, Dr. Strong transitioned to the role of assistant professor in residence in the School of Life Sciences. Dr. Strong's background is in HIV RNA structure and function with an emphasis on understanding how sequences in non-coding regions regulate various steps in the HIV replication cycle. She expanded on this work during her postdoc in the Schiller Laboratory, designing talons, a type of molecular scissor, to target and damage integrated HIV in cells. Currently, Dr. Strong, Dr. Sarkis, and Dr. Regner team teach the research-oriented undergraduate two-semester course Bio 207, Phage Discovery and Phage Bioinformatics. We're very pleased to have Dr. Strong with us today presenting about viral evolution, variation within virus populations. And from that point, I will turn it over to you, Dr. Strong.
Thank you, Corey. So I'm gonna click on this. We'll go entire screen, screen one, share, and then I'm just gonna switch over to this. We'll play from the start, and then we'll make sure we use, oops, actually, 2 seconds. I want you to be able to see the little pointer. So we'll play from start and make sure I get the pointer for you. That way we can emphasize a little bit more. Okay, fantastic. And Corey, once this pops up, if you could confirm that you can see the screen, I'd appreciate it.
Yes, I can see it. Looks great.
Fantastic. Okay, so you're here because we're dealing with a virus. It's a bit of a train wreck right now. And so if we want to understand this virus, we kind of need to understand viruses in general. And so we're all coming from different backgrounds with regard to our experience with viruses, our understanding of viruses, these different perspectives. And to make sure that we're all starting on the same page, I'm going to do a very brief introduction of a virus just to make sure that we all truly understand what we're talking about to begin with. So first things first, what you have to understand is single-celled organisms, multi-celled organisms, everybody's got a virus, at least one. And so the virosphere, which is how we describe all the viruses in the world, it is incredibly diverse. And so for humans, oftentimes we just get focused on what affects us directly. So for example, our top right corner, Ebola, or our middle, SARS-CoV-2, these directly affect our health and well-being. So we really, focus on these. We want to, study them, dig deep into the details. Now we can branch out from that. And again, we're very selfish creatures. If it affects us, we want to, study it. And so tobacco mosaic virus is an example where it was affecting agriculture. So, we must, really dig in deep and understand this because it's affecting us. Now, there is science for science sake, right? The idea we really want to expand our understanding of how things work in the real world. And that's where we start expanding out of just focused on us to other organisms, because other organisms are battling with viruses just like we are. So down below, you're seeing amoeba virus, and these are giant viruses. They're huge, but they infect a single-celled organism, the amoeba. So you have this giant virus that turns a single-celled organism into a viral factory. We've got bacteria that have to deal with viruses. So in the middle, you're seeing a bacteriophage. And this bacteriophage that you're seeing is actually a virus that was isolated by students in our Bio 207 course from an environmental sample. So they went out, got an environmental sample, purified this virus out, amplified it, and characterized it, and then they got to name it. So they named it Winston Payson. So, bacteria have to deal with viruses. And then if we're really, zooming out and looking at the way that life on Earth works, we like to classify it into domains. So you have domain eukarya, we belong in that domain. You have domain bacteria, so like E. coli, Staph aureus belong in that domain. And then the third domain that kind of sometimes gets ignored, that Dr. Hedlund in the School of Life Sciences studies, domain archaea, they have to deal with viruses. So in the bottom right-hand corner, you're seeing one of the viruses that targets a member of domain archaea. So really what I want you to understand is there are a ridiculous number and type of virus out there, and we just study a tip of the iceberg when it comes to these viruses. So, okay, great. You tell me there's all these things out there. What do they have in common? What they have in common is you have to have a way to get them from host to host to host. So you have to be able to deliver the goods safely. And what do you mean by deliver the goods? You need to deliver directions on how to build new viruses. And so what we see is the package that's going to be doing the delivery is known as the virion. So when we talk in scientific terms, it's very important that we use a proper vocabulary. So if you're talking about a virus outside a human being, that's going to go from person to person to person, you're talking about what is known as a virion. Within the virion, you have directions. So on the left-hand side, the directions are of a nucleic acid nature. So it's going to be RNA or DNA, depending on which type of virus you're talking about. So Ebola has an RNA-based genome versus human papillomavirus has a DNA-based genome. Now, okay, you've got directions, fantastic. Well, we want to make sure that those directions get to the next host safely. And so you have to have a way of packaging those directions and protecting them from the environment as that virion is tumbling, trying to reach the next host. And so there's different ways that you can protect those directions. The simplest way is what we call the naked virus, in that you just surround the directions with a viral protein. So lots and lots of viral proteins taking on a characteristic shape. Now, if we want to add a layer of complexity, what we can do is we can still have that layer around the directions, as we see on the top right-hand side, but then let's add another protective barrier in the form of a lipid envelope. So these are known as enveloped viruses. And so down below, you can see a really simplified depiction of what a naked virus would look like if you wanted to do a cartoon versus what it would look like if you examined it using transmission electron microscopy. So it's like, okay, we have a way of delivering the package from host to host to host, and that is known as the virion. And again, vocabulary is really important because when you don't apply the correct vocabulary universally, people get confused about what you're talking about. So an example of that is contagious versus infectious, and how those words are used differently in the public versus used within the scientific community. So if you're a virologist, you go, oh, the word contagious means it can be transmitted from person to person to person. You know, and if it's highly contagious, it's really easy to transmit it from person to person to person. If you're using the word infectious, however, when we use the word infectious, what we're talking about is how many virions, how many of those little packages have to get into the next host in order to actually establish an infection. So you don't want to use those two words interchangeably. One is getting from host to host, and the other is actually setting up an infection within the host. So you've probably heard about the R0, the basic reproduction number at this point. And here you're seeing a depiction of the how contagious different viruses are, depending on which virus we're working with. So MERS is a coronavirus that you may be familiar with, and so it's 0.8. So for every one person infected, on average, like if you averaged everything across, it would be less than one person gets infected by the original person. Influenza, it's 1.5, so for every one person infected, 1.5. You can see measles is a nightmare. For every one person, 16 people. When we look at SARS-CoV-2 right now, this is just based on the data that we have right now. For every one person, it's 2.5. Now, as we accumulate more data, this may shift because we can see that SARS-CoV-1, it's actually 3.5. So we won't know until we have more time to actually survey the populations what this number truly is for SARS-CoV-2. So transmission, going from one host to another host to another host. Well, if we look at humans, what we really focus on with regard to virus transmission is aerosols. So you can see on the right-hand side when someone coughs. The idea is you are forcing that air out and everything that's contained therein. And so they've done some really nice studies at Florida Atlantic University within their engineering department to depict this for the public, for them to understand the way that aerosols can disperse and carry viruses. And so for this particular image from their study, this particular cough went out to about 12 feet. And so there's a reason why that individuals have been asked to wear masks. I know that that's been very confusing with regard to don't wear masks, do wear masks, don't wear masks. And so originally when they said don't wear masks at the beginning of this year, the reason they said that is it won't prevent you necessarily from getting infected, and they were really, really worried about hospitals having enough of the masks that they needed. So the reason they ask you to wear a mask now is they don't want you infecting other people. And so it's like, well, how's that going to stop me from infecting other people? When you talk or you cough, those aerosols are going to stay more localized next to you, as you can see in the bottom right-hand corner. And so if it stays more localized next to you and you're also employing social distancing, the chances of you infecting someone else, especially unintentionally because you may be asymptomatic, those decrease. And so that's the reason why they would like people to wear masks right now is it's not to protect you from getting a virus, it's to protect other people from you, basically, if you were asymptomatic. So we have to worry about aerosols. For some viruses, you worry about food and water, so hepatitis A is a really good example of that. Fomites, so say you cough into your hand and then you touch a doorknob or you touch an elevator button, someone comes along and touches the same button and then touches their face, oh, they just contaminated themselves. Bodily secretions, sexual activity, so HIV is very famous for this. Birth, so you'll have mother to child transfer that can happen, transfusions like blood transfusions, and then organ transplants, and then zoonosis. So a famous one within the last few years was Zika. So the idea of mosquitoes acting as vectors
and delivering Zika to human beings. So these are all ways that viruses have been known to be transmitted among the human population. So, okay, we got transmission. What do we have to do if we're a virus? We're into the new host. Now what? So what you have to understand is that a virus is an obligate intracellular parasite. So what do these fancy words mean? Obligate means I have to do it. Intracellular means get inside a cell. And parasite means take over the cell. So get inside, take over, make more of myself. So how are you going to do that? Well, you need to replicate or make copies of your genome. So you need to have more directions made. You need to have a way of protecting those directions. So you need to be able to package them. How are you going to do this? Well, you got to convince the cell to alter its function. It needs to be focused on making you. And it needs to just stop doing what its normal process is and instead focus on executing my directions. And so when you get into the cell as a virus, there's so many cellular resources that you can take advantage of. There's amino acids and sugars and nucleotides and lipids. And all viruses have to get into cells because if I want more protein made, I have to have the ribosome make it for me. And so the ribosome is an organelle found within our cells that is our protein maker. It performs translation. So viruses, I need proteins. Okay, you've got a machine that makes proteins. Fantastic. Let's make this work. So I told you that with the virion, which is released out into the environment, you've got a blueprint in the form of nucleic acids, and then you protect the blueprint using proteins. Well, with virologists, we like to classify viruses. Like, how can we start putting these guys into categories? Because we're scientists and we're anal retentive like that. Let's put it in a box. And so David Baltimore, who won the Nobel Prize back in the 1970s for his work on cancer-causing viruses, came up with a system. And we still use the system today. And it's known as the Baltimore classification system. And the way that he laid it out back in 1971 was, let's look at what are your viral directions or blueprint? What is it made of? Is it RNA? Is it DNA? Is it single-stranded? Is it double-stranded? What's going on with it? So here, when he first proposed it, he only had 6 categories because we hadn't identified a member of the 7th category at that point. But then along came hepatitis B. So we got our group 7 viruses. And so there are 7 groups. And when I teach this, the virology course at UNLV, we actually go through all 7 groups and we have one representative virus of each of the groups so that students really work through it. So what we can see is that the differences between these different groups is based on your blueprint. So are you double-stranded DNA, so the double helix? Are you single-stranded DNA? Are you double-stranded RNA? Are you a single-stranded positive RNA or a single-stranded negative RNA? And then we'll see that group 6 and group 7 are kind of weird because you've seen the single-stranded positive RNA before. It was over here. So what's the difference between group 4 and group 6? Well, group 6 has this really cool protein called reverse transcriptase that does this really cool thing where it takes the directions and it reverse transcribes them into DNA, which is kind of weird. It's a cool backwards system. And so group 6 are very unique. HIV is a member of group 6. And then you also have group 7. So you had double-stranded DNA. You saw that before in group 1. So what's the difference? These have a really cool reverse transcriptase that they're also going to use in their viral cycle. So we've got 7 groups that we work with. All of them, one way or another, they need to come up with temporary messages known as mRNA that can be used by the cell, specifically used by the ribosome, to make them viral proteins. Because they need the viral proteins, 1, for part of their shell as they leave, but 2, also to be able to take over the cell. Because remember, I want to alter the purpose of the cell. The cell needs to focus on what I want it to do. Now, with regard to how the rest of this particular presentation is going to proceed, I'm going to go over some really well-known viruses first. So we're going to talk about HIV, and then we're going to talk about influenza. And I'm going to briefly touch on a relatively new virus, Zika, and then we're going to transition into SARS-CoV-2. So This work that you're seeing represented on the screen right now was the work that I did initially as a PhD student, where I was really, in the details when it came to trying to understand how HIV was manipulating the cell to do what it wanted to do. And so one of the ways that you do that is with HIV, it's got this RNA-based genome, and the RNA-based genome will fold up into all these cool different shapes, and the shapes are actually signals that can manipulate what's going on inside the cell. And so we wanted to understand how these different shapes come about, and then what are the consequences of these different shapes. And so there was a section of the genome that I was interested in that has nothing to do with coding for proteins. There are no genes involved in this section. It's what we call a non-coding region. And within this section, there was a region called the C box and a region that we called the G box. And so it's represented down below. The C box is just called C box because there's a whole lot of C's represented in that particular region. And then the G box is called the G box because there's a whole lot of G's. And so what we think was happening is that the RNA was folding back on itself, and it was really important that these two sections, when it folded back, made this connection. We thought it was really important with regard to propagating the viral cycle. And so we had proposed that this interaction is important, and that if you were to prevent the virus from being able to do this interaction, perform it, that there would be consequences that you would be able to detect doing experiments. And so I was like, okay, this is our hypothesis that this CGI is very important. And one way that you can test that if you're a virologist is you can build different versions of the virus and then say, okay, can you work with what I've given you so far? And so what I did was I built 3 different versions of the virus based on the C box, G box. So what we'll see is that in version 1, depicted as a little orange star, I took the normal HIV virus and I mutated the C box and I turned it into a G box. And so now I would predict that it would no longer be able to interact with the original G box, and that would be bad for the virus. I would predict that it would perform poorly. I created a second virus with the little blue star where it was like, okay, leave the C box alone. It can stay full of C's. Mutate the G box so that it can't base pair, interact with the C box anymore. I would predict that this virus would do terribly. And then do what we call a rescue, where I've mutated the C box, I've mutated the G box, but by doing this double mutation, I would predict that you could still have the interaction happening. It's just kind of opposite, right? It's opposite. You move the C box to the G and the G box to the C. And so I should be able to have a rescue effect. And then you test your viruses to see, did it actually work? And so what you're seeing on the right-hand side is a series of experiments that I did. And so when we are looking for whether or not a virus is performing well, you can do what's known as a RT activity assay. And so the higher the RT activity, the better the virus is doing. And so you have to have different controls in your experiments to make sure that you actually built the experiment correctly. And so my negative control on the far right-hand side is known as a mock. And so this is just normal cells that haven't seen any type of virus at all. And what you see is that there is minimal detection down here, but this is known as background level. So this is my standard of, you have to be higher than this. This is background. This doesn't count. It's imaginary, so to speak. And then we have our positive control on the right or left-hand side with wild type. And so what I see is that days post-infection, at 3 days after I infected the cells, RT activity is detectable. 4 days it increases, 6 days it increases, 7 days it increases. So this is my positive control. This is what I expect a healthy virus to do. So then I check my mutants. So my C box mutant, my orange one, terrible. Fantastic, that supports my prediction. Okay, good, my hypothesis is looking good. G box mutant, terrible. Fantastic, supporting what I thought, great. Rescue, terrible, terrible, terrible. So this was a very sad day in lab. And what I realized was, nope, I'm not very good at building viruses. Like, I thought for sure that this would be a rescue, it'd be beautiful, and it didn't work. So I went back to my PI at that time, and I said, my 5 nucleotide mutant didn't work. He's like, okay, scale back, just mutate 2 nucleotides, see if you can, similar experiment. Did that, didn't work. And I'm like, as a human being, I am terrible at building a virus. And so we were sitting there brainstorming, and he said, let's let the virus decide what the virus wants. So what I did was I created a library of viruses, a population of viruses. So that C box area, I did what's called randomization. I said, okay, you can be any nucleotide at this position. So N means you could be an A, a T, a G, or a C. G box, same thing. And so I built all of these different combinations and created this very, very diverse viral population. And then I said, it's time for you to compete. Out of this viral population, who can survive, who can thrive, who can reproduce, and show up in the next generation. And so you do this over and over and over again, and you force this selection based on these particular sequences. Which one is going to be the winning sequence? And so at the end, when you've gone through this, and this took months and months and months of pushing the evolution, what you can start teasing out are patterns. What are the patterns of success? And so originally, remember, I thought there was supposed to be this long distance interaction happening. And this is showing you actually how HIV can fold up into these really cool shapes. And so I thought it was this that you see on the left-hand side. I was partially right, but partially wrong. And that's why my original experiment didn't work. What the viral evolution ended up telling me was this part is nice and linear, but then this part needs to be able to branch. It's not supposed to be interacting with itself. So if you zoom in on those sections, again, completely linear, nice, this is what we thought on the left-hand side was happening. We were wrong. What was happening was what is depicted on the right-hand side. And so that's why oftentimes when we talk about, just why can't you solve what's going on with viruses? Well, we think we know things, and then we realize, no, you really don't know what you're doing. And so it's important that we pay attention. Now, you can see we generated a variety of these viruses, and they were all competing within this population. And so there was a suggestion that maybe, by competing with one another, some of them are playing off one another to be winners, and some of them are playing off, one another and just losing. And so we're like, okay, let's pull some of the winners out, pull a loser out, and then let's see by themselves, how did they do? And so we have wild type as our control. We expect wild type to do solid, do very, very well with viral production. And then I had 3 winners that I pulled out, 119, 120, 121, depicted as the white, the light blue, and the darker blue. And then I had my loser that I lost really early on in the evolution experiment. I saw this virus initially, and then it disappeared. It wasn't represented in the population anymore. So I would predict it should be a terrible virus. So what we do is we compare them. And so we see wild type depicted on the right-hand side, second from the right, nice, solid, viral replication as depicted by the amount of capsid protein, the protein found in the virus particle that's being released. Okay, you're doing well. You're producing good virus. The loser is terrible. Great, fantastic. You are a loser, and you're still a loser. And then the ones that I had predicted would be winners. Okay, this 119 on the far left-hand side, you're still good. It's not fantastic, but you're a solid performer. The light blue 120, you're good. You're doing solid work. And then 121 was actually initially better early on in the viral cycle than wild type. So it was like, we had a little bit of gain of function going on there. And we ended up doing follow-up experiments to determine why that was. And again, with science, one outcome leads you to come up with some questions that leads you to more experiments. So that's what's really great about science is it's going to lead you into more science, basically. And so what we see here is that this is what was happening in lab. I was looking at just a few nucleotides, right? So look at that. We got 6 nucleotides here, 6 nucleotides. So 12 nucleotides out of 9,000. had this effect. And it wasn't even in a region that most people would think would be important, because it's not a region that codes for protein. And so, you have to think, this is what's happening in lab. What do you think is happening in the real world? So these viruses, these populations that you find in organisms, are constantly having to evolve. They're constantly having to deal with, pressure from their hosts, right, fighting back. And so one famous example with HIV of where the host fights back is there's a host protein known as AppleBeck that says to HIV, no, not in my house. You are not establishing your infection in this cell. So there was a research group, Harris et al., that was like, okay, AppleBeck is a cellular protein that says HIV, it's not happening. HIV population has evolved such that it produces a protein that goes, yes, it is. My protein is going to mess with your protein. They're like, well, what if we take away HIV's weapon? We take away that protein. Can HIV figure out a second way to get around this system? So when I was talking about, this relationship, what I'm talking about is on the left-hand side, you're seeing in purple is a cell. The cell has been infected by HIV. We're making brand new HIV particles. And there's this host protein in green, the little green circle known as AppleBeck. This protein gets packaged in the virion. So the virion, it's moving to the next cell, and it's going to infect the next cell. At this point, AppleBeck's inactive. It's not doing anything. It's not going to be active until HIV tries to get established into the next cell. Because what happens is HIV has to go through some initial steps in order to get established. It needs to take its RNA genome and convert it into double-stranded DNA. And the way it does that is using the enzyme reverse transcriptase. Well, AppleBeck's like, no, I'm going to mess you up. And so what it does is it induces mutations in the directions that reverse transcriptase is trying to use to build the proviral DNA. And it creates so many of these mistakes that the genome ends up just worthless. It's just absolutely worthless. And so what has happened is over time, HIV has evolved as a population such that they produce on this right-hand side this protein called VIF. So this little green circle right here, this is an HIV protein. And when it encounters the host protein AppleBeck 3G, it goes, come here. There will be none of this. You will not interfere with us. What I'm going to do is I'm going to push you through this pathway, and you can see it's a pathway indicated by the arrows. And at the end of the pathway, what ends up happening is the cell ends up destroying its own cellular protein. So AppleBeck ends up just getting torn to pieces by the cell because this HIV protein tricks it into doing that. And so they're like, okay, well, what if you don't have this trickster around? What if we stop HIV from being able to do that? Can it figure another way around AppleBeck? So what they did was they created initially a viral population that could not make that VIF protein. It was unable to. They mutated it so it can't. And then what they did was they grew it up into cells that did not have any of that AppleBeck present. And then they did some selective pressure. So they're forcing evolution in a test tube. They're like, let's add a little bit of AppleBeck expression, make a little bit of that AppleBeck protein in those cells. And then let's make a little bit more. Let's make a little bit more. It's like a whole bunch more. So you give it time to adapt. So the same thing with regard to antibiotic resistance and antibiotic resistant bacteria. You can get resistant viruses if you give them enough time as a population to evolve. And so at the end of their experiment, they're like, do we have any adapted viruses that have worked around this AppleBeck system? And what they found was, yeah, they had 3. So on the left-hand side, they've got isolate A, isolate B, and isolate C. So these represent the 3 viruses that were winners at the end of their experiment. And so depicted in the middle where you see the long gray bar, that's showing you the mutation series that they put into the VIF gene to prevent it from being made. And notice that it's kept. So you're still not making VIF. So it was like, okay, it didn't mutate back to have the ability to make this protein. Okay. So, where are these mutations? How do we think they're important? So the mutations on here are depicted by the vertical lines that are going through the long horizontal line representing the genome. And there was an area of interest down here where they've highlighted in light blue and orange. The envelope proteins, these are the proteins that are depicted on the outside of the virus. And they're like, why would proteins on the outside of the virus matter when it comes to the inside? So they're like, we really want to investigate these mutations, but we don't want to be confounding our results by not taking into account these other mutations that we're not as interested in. So what you do is you build versions of the virus that don't have all these other mutations you don't care about. They only have the ones that you do. And so when you build these, you have to actually make sure that they work. And so that's what they're showing in D, is they wanted to show that their envelope A mutant, their envelope B mutant, and their envelope C mutant all were capable of making virus. And they were, so they're like, cool. So they do a series of experiments to try to tease out what is going on, why would you have these mutations in this outside protein? It's just weird. And what they determined is the coolest system. So up top is depicting what normally happens, where you have a virus established over here in the top left-hand corner. You end up generating more more virus, and you have to worry about the little black stars, which are Applebeck. So Applebeck ends up getting packaged. Here we're showing a 1-to-1 ratio of 1 Applebeck for every reverse transcriptase. Remember, that's who Applebeck messes up. It messes up reverse transcriptase. So here you see a 1-to-1 ratio. And so this is a bad situation for the virus. When they did those envelope mutations, what they see down below is the way that it gets around it is, I'm going to put more of that reverse transcriptase enzyme into each virion. So you're targeting my worker. Well, I'm going to put even more workers into the virion. So by the time you get to the next cell, you can try to do this 1-to-1 targeting. It's not going to work because I got double the amount of reverse transcriptase in this virus particle. So you're going to have to try to play catch up with me. So this is kind of cool because they would never have predicted that, that somehow the envelope protein is leading to more reverse transcriptase being packaged. So it's cool that you let the virus decide, well, how are you going to work around this system? Because then we learn new things about the flexibility of these viruses. So here, this is all cell culture. It's great. But really, if you want to scale up and be more physiologically relevant, you need to go into an animal model system. So like with influenza, bird flu, the biggest issue with bird flu is they were scared that, is it going to become very efficient at passing from human to human? And so they're like, okay, we need to do some experiments and figure out what to look for with regard to viral surveillance to see if we can prevent this from happening. So big picture justification for this study by Fouchier was, can it acquire the ability to be aerosolized and pass from mammal to mammal to mammal? So ferrets are fantastic influenza systems, so that's what they're using as their model system. And so here, what they really want to do is they didn't want to study genetic shift, which is where 2 viruses simultaneously infect a cell and swap parts. Instead, they wanted to look at drift. Could this virus population just over time accumulate mutations that allow the virus to be passed from ferret to ferret to ferret to ferret? So what they tried to do was they tried to build the virus. And like me, they're not very good at it, and didn't work. So then they realized it didn't work. Let the virus decide. So what they were looking at initially was they were trying to design it based on receptors, because it's like, oh, cellular receptors are so important. The preferred cellular receptor for bird flu is what's known as alpha-2/3. For humans, it's alpha-2/6. And notice that my little ferret's got alpha-2/6 and it's also got alpha-2/3, as do humans. And so they're like, we're going to give the bird flu the right receptors that have a preference for 2/6. It's going to work beautifully, and it didn't. They were wrong. So they're like, we stink at this. What we're going to do is we're going to force it to evolve in the mammalian host. So they took their virus, and then they stuck it up the nose of the ferret, and they let it incubate, let it make virus particles, and then they took a sample from ferret number 1 and stuck it up the nose of ferret number 2. Did the same thing, stuck it up the nose of ferret number 3. So they're physically infecting these animals. Boom, 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 all the way down to ferret number 10. And so this took time. And what you see here is that in yellow, on the left-hand side were the original mutations that they thought were going to be key to being able to transmit from animal to animal using aerosols. Those mutations were kept. But look at all these other mutations that were acquired. Okay, so cool. Now let's see if we can get the ferret to pass it to the other ferret without us interfering. So the way that they did that is through an aerosol transmissibility test. So you have ferret on the left-hand side and a ferret on the right-hand side, and what's separating them is a cage. And you have the air direction moving from left to right. And so they would inoculate the ferret on the left with the virus and then see if, through its aerosols, it could infect the ferret on the right. And it worked. So they took that virus, stuck it up the nose of a ferret, and said, hey, can you pass it to other ferrets through their cage? Yes, 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 no. So 3 out of 4, that's pretty good. So then, they're showing you the different mutations that were identified in these samples. And they went, okay, ferret number 2, you're making a lot of virus. We're going to take that, we're going to grow it up, we're going to inoculate a new ferret, and we'll go, okay, ferret, can you pass it to other ferrets? Yes, 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 you can. Cool. So then they said, okay, ferret number 5, we're going to take your sample. We're going to get a very fresh new ferret, stick it up its nose, and put it next to some naive ferrets and say, hey, can you pass it through the air? F7, yes. F8, no. And so then they went back to all these original viruses and did a comparison of their genomes and said, hey, what's important? What's not? And they found 5 key mutations. And they wrote it up and they submitted it to Science. And there was another group that did very similar kind of sets of experiments, and they sent theirs to Nature. And this freaked everybody out at those different publications because they're like, these are gain of function experiments. I don't know if we can publish them. And so it became a very big thing within the scientific community and within the worldwide system with regard to governments getting involved. And there ended up being a moratorium on this type of research because they thought it was very, very scary. Now, the researchers were like, okay, we show that it can be passed from ferret to ferret to ferret, but you really need to take into account when it's in the airborne version, it doesn't kill the ferrets. So it's not deadly. So when they did this series of passaging, it attenuated the virus. So it's no longer deadly, but it acquired the ability to be passed from ferret to ferret to ferret. Now, if you changed your inoculation route, if you just jammed it up their nose or down into their trachea, you could kill some ferrets. But just the normal, natural spread through aerosols, this virus, it didn't kill ferrets. So they're like, please take that into account that it's not resulting in death. We didn't make a super scary monster virus. Instead, we made a virus that's capable of being, passed from mammal to mammal, and now we know how. So then when we're doing our viral surveillance of influenza throughout the year, we can look for these types of mutations and be on guard. Still, there was a moratorium on this. This publication came out in 2012. And it wasn't until 2019 that it was, they've actually lifted the moratorium. So it's still very controversial, but it's continuing. And this is using a system that we're very, very comfortable and familiar with. So imagine using systems that we're not so comfortable and familiar with, such as Zika. So with Zika, what you got to understand is in the scientific community, we knew about Zika. Zika was discovered in the 1940s. And there were researchers that were looking at it, and they knew that it was transmissible sexually. They actually wrote a paper about it in 2011, where a researcher accidentally gave it to his spouse through sexual contact. So it was known that it was transmissible. But with viruses, it's not a problem until it's your problem. So at this point, Zika wasn't on really anybody's radar. And it wasn't until 2015, 2016, when you started seeing the microcephaly occurring within South American strains that people got scared. And so it was like, okay, now we need to dump a lot of money into studying Zika. We need to understand what's gone, why is it doing this? Why is it causing this? And some really great research came out of it, where they figured out that within the fetus, as the embryo was developing, you have a lot of crosstalk that happens between cells. And so certain cells here depicted in yellow that are infected with Zika, the chemicals that they're releasing to communicate with other cells, they're releasing unnatural levels that you do not normally see happening during developmental stages within the fetus, and that is messing stuff up hardcore. So one way they were able to demonstrate that is they're like, okay, let's show you with neurospheres how development should look normally, and then let's show you if we add signals at the level that Zika is causing cells to secrete, what happens to the developmental process. And so you can see the LIF is one of the signals and the VEGF is the other. And if you just do a comparison of the control on the far left and what's happening in the middle and the right, you are really messing up this developmental patterns, these interactions between cells. And they're like, you're messing up communication. Their cellular communication is getting just really disturbed. Now, why? What shifted between Zika from the 1940s, 60s, 70s, 90s? Because it's been endemic in the continent of Africa and Southeast Asia for quite a while. So what shifted? And so a group did some genomic studies trying to do comparisons to see what shifted. And so with Yuan et al., what they found was they pulled some original virus, Cambodia 2010 strain, and they did a comparison with the newer strains using a neonatal mice system. And what they found is that the Cambodian strain really, you would lose about 20% of the mice, but with the newer strains, it was just killing these mice. And they're like, so there's this neurovirulence difference between these different strains going on. And if you look at the little brain buckets, you can visibly see differences. So MOC would be your typical normal size brain. The Cambodia 2010 strain infected versus the Venezuelan strain. And you can compare the red lines, how far out they extend. And you can see these brains are much smaller. They're not developing the same way that the others are. So they're like, okay, that's a problem. We see a difference between the 2010 strain and the 2016 strain compared to normal brains. And so then they did the genomics comparison, and they're like, well, what's different? What's going on? And they're like, well, about 2014, 2016, we start seeing this particular change in an amino acid in this PRM protein. And they're like, we wonder if that's really important that this particular mutation is showing up. And that's when we start seeing all these microcephaly cases being recorded. And so they built viruses that had these unique mutations, just these mutations compared to the original Cambodian strain. And what they found was just this one difference between the Cambodian strain depicted in black and the new mutation depicted in red. It dramatically dropped percent survival of those mice. And they're like, this looks very, very important. They're like, well, can we rescue it? Let's go back to the Venezuelan strain, which just slaughters the poor little mice. Let's just make that single reverse mutation. Let's turn it back to what is originally found in the Cambodian strain. Can we rescue? And they could up to a point. So they ended up having 40% survival, which is better than 0%. So they're like, wow, this single mutation doesn't account for a portion of what we're seeing with regard to this neurovirulence. It doesn't account for all of it. We're still losing 60% of the mice, but this mutation seems very, very important and we should pay attention to it. Now, the problem with that is that, with scientists, it's like, oh, it's important, but it's not the only thing. But sometimes what happens with science is people take that information and they kind of blow it out of proportion. And so when they were having outbreaks of Zika in India, part of the government going forward is they didn't want travel restrictions to India to occur. And so they screened all the genomes that they were pulling from people. And they said, oh, look, we don't have that S139 mutation, so it's safe, you should come. It's like, no, that one change, it contributes, but it's not the only contribution that's happening to get microcephaly. You can't base your decisions on this one mutation. That's not good science. And so here you have this misperceived idea of risk because things are not being interpreted correctly. Now, if we get to Rona, I'm going to quickly finish up with Rona. What you have to understand with SARS-CoV-2 is we're out 6 months. We've known about this virus 6 months. That's it. And so we're scientists working very, very hard to try to figure this out. There's been some good work trying to determine where the original source was for this virus, looking at doing genome comparisons of certain sections of the genome. It looks like sometimes there's a little pangolin in it. So bats are fantastic for acting as reservoirs for SARS, but oftentimes there's an intermediate. Looks like there's a little bit of pangolin, but not just in this section. So it's a really weird looking virus. And so they really haven't determined what the intermediate is. And so there's going to be a lot of work that's going to be needed to do this, right? Because it's a scary virus. We need to figure it out. We need to prevent additional outbreaks from happening. Now, the US had understood this back in 2014. We had funded a grant on this, understanding the risk of bat coronaviruses. And they had just produced fantastic information. And so here, I mean, with regard to their papers, they were just publishing like 18 peer-reviewed papers and really, 2 in Nature, 1 in Cell, really good journals. They were generating a lot of data, came up for a grant renewal in 2019. They're like, absolutely, yes. What you say that you need to address, such as the origins, diversity, capacity to cause illness and risk, absolutely, we need to fund this. And so it was funded. And then we had COVID-19 outbreak, and NIH pulled their grant because of some political influences. And so that was very concerning to a lot of the scientists, because it's like this stuff is really important. It's really important that you have an international collaboration happening between scientists all over the world. So this one happened to be between scientists in the US and China, and they had established this collaboration. It was 15 years old. This was a long, strong collaboration where they were sampling bats and collecting, all of these different samples in order to try to catalog this so that we can, have feet on the ground, figure this stuff out, and then you just cancel it. So that's very dangerous, because we need to move towards more of a collaborative nature, which is what a group from Harvard has said, is we need to start working as collaborators. We need to start surveying earlier. They'd like to set up a global immunological observatory where they're taking anonymous blood samples from blood donations, plasma donations, what have you, and they're keeping their eye on it with regard to antibodies, with regard to infection versus you've been exposed and now you're recovered versus you've lost your immunity. And they come up with ideas of how could we actually make this work. And so with that, I'm actually going to stop, because I know that I went really long, and I'm sorry, Corey. So I'm going to stop with that.
You're fine. Thanks so much, Dr. Strong. This has been awesome. It's a fascinating presentation, and there's so much information that you gave to us. So we really do appreciate you taking the time to do this and put this together. I know it's a lot of work, so thank you.
You're welcome. So questions, hopefully people can still be here to ask questions.
If there are any questions in the chat, there's still a good number of people with us. So if you have any questions, pop them in the chat. I'll wait for a minute or two. I didn't see any come through during the presentation. But if there are any questions, give everyone a minute or so. Doesn't look like there's any coming through. And oh, here's one. Do we know if scientists are working towards getting the NIH grant back?
They are. This is an unprecedented time. They don't, as far as I know, within the records, this has never happened before with regard to political influence in the United States pulling a funded grant. So they are trying to work towards it. I don't know if it's, the legality of it is in question, whether or not the NIH has the right to do it based on the criteria that they submitted. Because originally, you can follow the timeline of where the grant was discussed within the political arena, and then 10 days later, the grant was defunded. So it's very controversial. Scientists are very scared that, hey, wait a minute, you can't, you're making policy decisions on scientific grants that have been vetted, that have worthwhile, value.
Then,
so, it's very. So with the political influence, with that, it's the connection to the Chinese researchers, and so the collaboration was with a lab in Wuhan. And so this, again, it had been 15 years, but I think if it wouldn't have been in Wuhan, China, I don't think the grant would have been pulled. But because one of the collaborators was based in Wuhan, China, yeah, if they had been based in South Korea or Japan or Australia, I don't think that necessarily would have caused that grant to be pulled.
There's another question on that from Monica. It says, how contagious and infectious is COVID-19 based so far on data?
So contagious-wise, a little bit more than influenza. Infectious, we don't know. So that they're going to have to tease out the infectious results. Now, what's interesting is they found that individuals that were working in mink farms. So minks are like these little. They're related-ish kind of to ferrets, but you have mink farms in the Netherlands. They found people were accidentally infecting the minks, and then the minks were going back and infecting new people. So I think we're going to have to do some animal model studies where they are really controlling how many virus particles to see, like within a mammalian system, how many virus particles is it going to take? Because like norovirus, it's like 1 to 10 particles is sufficient to give you what's known as colloquially the stomach flu. And so that is incredibly infectious. While like Ebola is not, it's not highly infectious at all. It's highly contagious, but not infectious.
Interesting. One more question, it looks like. So in the context of evolution, can you talk about finding stable targets for developing vaccines?
Yeah, with regard to the vaccines, it becomes a little challenging because oftentimes what we target is what's displayed on the outside of the virus particle, because that's what your immune system is most likely to encounter. So it's usually like the envelope proteins. And depending on the virus type, viruses can have that mutability, that ability to over time accumulate mutations. Now, if you are an RNA-based virus like SARS-CoV-2 is, oftentimes what happens is the protein that's responsible for making your genomes that are going to give you your proteins, it makes mistakes and introduces mutations, and then so if you're targeting a specific part of that protein, it may change within the population, and then you select for those that get around it. Now, they're doing some really nice studies with neutralizing antibodies that they're pulling from patient serum. And what you do with that is those neutralizing antibodies are really good at binding to the virus. So you try to map out what parts are they binding to, because maybe those parts are what we call conserved. They're less likely to be able to change. So then you can design your vaccines around that. You want areas that are less likely to mutate. So like with HIV is notorious for mutation, and that's why it's so challenging to be able to design a vaccine against it.
Interesting. There's one more question, and then we'll have this be the last one. And it's, is there a difference in severity of disease based on what organism the virus may have come from? Why is finding the animal origin of the virus important?
It can be, because it usually seems like with viruses, oftentimes when it first transfers, when you first have that transfer, it doesn't go well. It does not generally go well. So like with Ebola, when Ebola first got transferred from the bat reservoir to humans, it was just lethal, right? And so people, when they think of Ebola, it's like, oh my gosh, you're just going to die from it. But what's happened over time is you kind of have that back and forth, and now there are different versions of the Ebola, different strains that are not as deadly. And so it's establishing relationships, basically. So like with HIV, where it's cousins to what's known as SIV, simian immunodeficiency virus. And SIV's been in the monkey population for a ridiculous amount of time. And they've developed a relationship where they can carry high loads of this virus and not have any problems. They've developed this relationship over time, right? So the longer you're together, the more potential for attenuation. It's like, okay, you're not going to, because if you kill the host right off the bat, your chances of being able to spread decrease because you killed the host. Versus the longer you can have a relationship with one another, the more potential for spread. So it's not that, technically, viruses are not alive, and they don't have a ultimate set of goals. But in the long run, those that are going to survive, thrive, and reproduce the best are going to want those that can be propagated most effectively. So the longer you can stay, the more effective your propagation.
Interesting. Also fascinating. Thanks so much, Dr. Strong. We really appreciate you taking the time to do this. As I mentioned at the beginning, you can check out the recording. This one should be up hopefully within the next week, but it will be at unlv.edu/sciences/virtual-lab-notes. You can check that out there. And then also remember that we do have another virtual lab note session coming up on July 29th with Dr. Ernesto Abel Santos. And then the topic for that one is prevention and treatment of infectious diseases in the COVID-19 area and beyond. Thanks so much, everyone, for joining us today. Thanks, Dr. Strong. We appreciate your time. We appreciate you putting this together for us.
Well, thank you for having me.
All right, I'm going to end the recording, and we'll hope to see you guys on the 29th.